Hi, this is Roger Green, host of the Surfing the NASH Tsunami podcast. This weekend, we are offering 4 conversations from Season 3, Episode 45, our review of the recent Paris NASH conference, plus, from the vault, conversation from April 22, which looked at the need to improve testing criteria for drug development. These conversations are running a bit behind our usual schedule due to the challenges of travel and time change surrounding the NAFLD summit that ended in Dublin on Saturday. We'll be back on our regular conversation drop schedule next week. And now, on to the discussions. This conversation starts with me commending the other talks in Session 5, the session that Frank Anania started. Roy Sabo presented a cogent, comprehensive discussion of the value of adaptive trial strategies, and the following 2 talks discussed organizing patient databases in ways that simplify trial recruitment and have the potential to reduce screen fail rates. In the last talk, from Marcus Hompesch from Prosiento, we discussed the pluses and minuses of educating patients on better self-care. Good news, it creates a larger ready-made patient pool and improves overall patient health. Less good news, it increases placebo rates in clinical trials because patients are better informed and provide better self-care, which requires drugs to perform better if they are to demonstrate statistical difference. I didn't call this bad news because we should expect better performance from drugs in a world where better informed patients manage themselves more aggressively and appropriately. And from there, the group goes on to discuss the rest of the Day 1 program. Every year, Paris NASH provides some of the strongest scientific content found in any program, coupled with an innovative look at fatty liver disease in the context of world health. This conversation touches on what emerged as some of the high points of a fascinating and important meeting. So, sit back, listen, enjoy, learn, and when you're done, join the dialogue on our LinkedIn discussion group. We've talked about Frank Anania's talk, but we didn't talk about the rest of Session 5, which I thought was fantastic. Because what Session 5 took a look at was organizing to think creatively about better ways to do the important things we have to do in clinical trials. Roy Sabo, I've always understood at a 30,000-foot level that adaptive as a concept was a good idea, but I didn't really understand what they were talking about until Roy Sabo walked us through all that. And I thought that was amazing and very, very clear. I mean, Stephen had talked about a month ago about taking FibroScan reads midpoint and using that to reset an entry criterion, but this is a whole different level. And I think that the wizardry of, I'm a big fan of Bayesian statistics, the wizardry of Bayesian statistics strikes again, but I thought that was excellent. The talk after that was the fellow from Mexico about how we're organizing things in Mexico that I thought was really fantastic, Jeff, in terms of organizing around trial populations and how to get people more coherent to do that. And then the last talk was Marcus Hompesch, I think is his name, from Prosiento. I've been looking at Prosiento for 2 years, ever since they started raising money, as a really interesting model of how to attack clinical trial more systematically. Louise and I were texting during his talk trying to figure out exactly how far they're bringing the screen fail rate down, but it's a big number. And they're doing that by taking, I think, a much more holistic approach to what they're doing. You're on the kind of thing you've been advocating for in recent episodes, and you and Nail and IT and all that are kind of pushing towards as well. But I became imbued with hope that we can start doing trials much smarter, even than we envision today as we look ahead.
Jörn Schattenberg.
Session 5 was good. As a clinician, you're always a little bit intimidated by a statistician who gives you a lot of formulas and you're really trying to wrap your head around it. A lot of people felt a little bit overwhelmed by the formulas he presented. I forgot you're a statistician yourself, Roger, so you felt very comfortable with the formulas, but I could follow him, at least his words, and for sure the adaptation and being able to shorten or change the trial or enrich its power is clearly something we have to do in such a disease where we don't have an approved therapy, and then submitting people to potentially ineffective or highly effective treatments to shorten treatment duration. But coming to Marcus Hompesch, and it's a German name, so I would call it Hompesch.
Thank you. For those who don't know, Jörn teaches me European names from time to time, but I completely foul up. So this is the first time I believe we've ever done this in public.
Yeah, sorry for that, Roger.
No, don't be. Please, I need to learn.
And Marcus, we had an exchange in German, so I'm pretty confident that's all. You know, the question I had is, and I asked him that, is of course it's good to enroll the patients, but it changes things for the good. I mean, patients are going to reflect about their condition. Potentially those programs also provide something, access to either healthcare or educational materials. So there is going to be, maybe we're even going to be able to shave up some of the placebo effects that are normally then just start at the beginning of a clinical trial. And as such, I believe that these type of programs that enrich patients, keep them at bay to be enrolled once the right trial comes along is a good way also to decrease placebo effects.
Yeah, I think these are all enrichment strategies. And Bayesian priors as kind of not cutting edge, but certainly state-of-the-art state-of-the-art statistics go. Bayesian priors are a place to look for enrichment strategies without going into any details beyond that for now. Jeff, you wanted to say something on that?
Jeff McIntyre.
Yeah, I mean, it points, again, more towards patient involvement. And I feel like I'm becoming a little bit of the annoying parrot on the shoulder that only has the one phrase that he comes back with all the time. But it seems to me that this points to this. I fear that in many of our how do we improve clinical trials conversations, we talk about screen failure rates and other aspects of it, so often there is technological response of some sort. That if we can do this, if we can integrate AI, if we can integrate a different aspect of technology, that it will help incorporate that. And I think that I rank technology probably down at about 7 or 8, actually, of what I would consider priorities in terms of being able to recruit better and to get better results in clinical trials as well. There are huge inequities in the system. There are biases in how things are chosen. There's just a ton of other places that we can probably do that. And I think with patient input earlier on, it's going to help with that. I mean, even in the area of, frankly, something that wasn't discussed at Paris NASH, but informed consent, that plays with the notion of education to begin with. But there's a lot of work to do that will happen outside of the labs to be able to include to improve clinical trials.
If I'm being defensive, forgive me. I don't disagree with anything you just said. I think there's a question of getting the right population in where we are woefully deficient right now. And then there's a question of taking the population that's in and enriching it to the best end in terms of results. And in terms of enrichment, I think statistics has a lot to offer. In terms of getting the right patients in, I completely agree with you. And if you start with a population that's too narrow and you enrich it, which is the easiest thing to do, we're still going to wind up looking people in the eye in 15 years and saying, well, we haven't done any work with your particular ethnic group, but trust it, that's unqualifiedly bad. On the other hand, getting in populations that are appropriately diverse and then not understanding how to take advantage of the differences between them will produce trials that are inefficient as well. So you're right. If I had to pick one, I think I worry about the one that you're worrying about. Happily, I don't think we have to make that choice. I think we can get the different people to go down both paths and make the whole thing work better.
Yeah, agreed. I mean, not to be trite, but something's better than nothing. And if we're not maximizing those that are coming into the trials now, then we're not going to have a foundation when we try to expand as well.
We have a little bit of time left. I thought maybe we would just take the program and go session through session and see if anybody had anything they wanted to comment on that we've not touched on yet. Let's start in session 1, right, where certainly Louise has talked significantly, and actually we've talked about all that a little bit, haven't we? Is there anything anybody would like to add on the any of the discussion around session 1?
Something that stuck with me was that colleague Sanat from Friends motivated us to look at outcome trials and look at different indications. And I think this is something that the field needs to be discussing.
Okay, session 2. Well, actually, we haven't said anything about Chris's paper, have we?
We haven't, but I would like to start with Cyril Kasi. She highlighted different types of insulin resistance or type 2 diabetes in patients with NAFLD, and painting just 2 subclasses. One's with severe insulin resistance that over time develop NAFLD, and those with insulin and those with NAFLD that have some insulin resistance in type 2 diabetes, but maybe not the worst one. And I thought a very pragmatic and simple way to look at them, one or the other. And I thought that the treatment of these 2 types of diabetes are going to be very different. And actually, that was one of the questions that came from the audience too. I think we're not equally paying attention to the length of the diabetes or the quality of the control, and I think we're seeing subgroups here.
Spoiler alert, Jorn, which is that if she's in Dublin, I intend to beeline her and ask her to come do an episode on that, because I thought Louise and I both agreed as we were listening how fascinating it was. And if we can get her into the diabetes series also, that would be fantastic for the same reason. I agree. I thought that was remarkably good and clear data.
In the endocrinology literature, there's some more subtype. Lancet endocrinology paper described 5 subtypes with different organ manifestations. So I think there's more complexity to that. She did it very pragmatic. Maybe it'll be great to have her in an episode and detail it.
Louise Campbell.
But I did enjoy Chris Cowdery's session. That was the first one. His point was when we do randomized controlled trials, we get in a population that doesn't reflect the real world trials, which one's heterogeneous and one's homogeneous, and all of the problems that go on that sort of problem. Therein lies part of the issue of where we stand currently. We can have a perfect drug for a perfect heterogeneous patient in a trial that doesn't work in the real world. And I think, let's see. So he was talking about multiple different markers and how you do that. And I thought that was interesting.
I thought Chris is a great choice for that paper. He's been doing stuff for a long, long time, and he's not solely about fatty liver. He looks more broadly at liver disease. And I think the more broadly you look, the more you can appreciate the shortfalls that you run into into by focusing too narrowly, and I thought he did a great job of summing that stuff up. Louise, I think you were the one who pointed out before we got started the HIV paper. I don't know, was that when you stepped out, or were you, or did
no,
you
I caught some of that, but not enough to really be able to comment more than there was a general feel that a lot of people now with the HIV, and Jean is far more qualified to do this with his studies, is that now they gain weight the same as anybody else. And there was one argument that most of their NAFLD was related to their weight gain and the normal parts of life they now have. That was the only bit that I was able to catch from that session.
Bjorn, do you have any reactions? I know, obviously, as Louise pointed out, and we've all discussed, you do a bunch of work in this area.
They linked some risk factors that are present, medications, viral aspects, CD4 count to disease manifestations. Something that's always very striking to me is that in general, this population is a little bit younger, but the metabolic burden is the same. I think one of the reasons why they're so studied intensively is because they tend to show up in clinic regularly, and it's particularly easy to enroll them in such a preventive strategy to inform them about their health and to do it more globally, because it's a population of chronically diseased or chronically infected humans. As such, we spend a lot of time with them and counseling them. Metabolic liver health is just one aspect of that too.
Moving onward, as this clock moves on as well, Scott Friedman's session, I just thought fantastic, but go ahead.
A very special session. He always complains that he hasn't had that talk when he starts it. It feels like, he's been thinking about that for some long time, and the diversity of the methods and the papers he presented was just great. It's stimulating, and he highlighted some of the technologies that we also discussed in this podcast. Single-cell sequencing repeatedly said that this was the most powerful technology advancement he's seen recently, and I think we're going to see many more great discoveries from that.
He's been clear about that. He's been clear about his fit. So in my old career, every once in a while, I got asked to give a paper about something I didn't know anything about, and not so different. I think what you realize, is that everything you know ties into everything you need to study more than you would have realized when you get started, and it winds up shaping the perspective that you bring to the talk. I think, as I said, I could watch him read a phone book because I'm sure he would manage to bring all his experience to it, but this was an example of that. Jeff, you look like you want to say something. Do I have you wrong?
Dr. Friedman's, session was interesting. As you marked earlier, I'm in this new position now and am learning much more about HCC as I now oversee, see if I'm how many letters I'm throwing at you, GLI's liver cancer program. And it's interesting to make the connections there to see where the outcomes are, but also see where there are some dissimilarities as well. Just a good basic overview, for me of that.
So stepping into this role, was there any particular point he made that struck you, or was it just kind of in general, it was quite impressive and enveloped?
Yeah, that it's impressive and that there's still quite a long way to go with it.
FNIH, I'll let somebody else comment on that first. I learned a lot. I guess I had expected it might go a little deeper than it went, a little more about here's what we're doing and a little bit less about here's what we're learning. But maybe it's premature for that, and I was encouraged by the programs.
I would agree with that, Roger. It's an open secret that, the Liver Forum did its meeting the day before Paris NASH, and where we got a preview of a lot of these topics as well. And I'll find out, I guess, that, Frank Anania was a part of those discussions as well. And I feel like that, as we said earlier, really kind of steered a lot of the talk and a lot of the reaction we had to this. And so it was nice to see the FNIH presentations occur kind of outside of that perspective, kind of being a part of it.
And now, back to Roger.
We hope you enjoyed this recording. If you have any questions or comments about the content of this conversation or the entire episode, please send an email to questions@surfingnash.com. We will be back next week to discuss some recent exciting drug trial results and what they portend for the next couple of years in drug development. In the meantime, stay safe, surf on. We'll see you on the podcast. Bye-bye now.